The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of deoxyribose sugar, a phosphate, and one of four nitrogenous base. The phosphodiester backbones consist of alternating sugar and a phosphate groups. The nitrogenous base includes cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5 to 3 prime direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3 to 5 prime direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with the double-strand DNA being separated, and each original strand, called parent strand, is used as a template for the complementary base pairing of nucleotides to make two new molecules. DNA replication occurs in the 5 to 3 prime direction, adding new nucleotides to the 3 end of the newly formed strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalase the addition of new nucleotides to the growing daughter strand. Other proteins such as beta clumps and the clump loader help load the DNA polymerase in place on the DNA. Short sequence of RNA called primers have to be paired to the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occur at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs in the 3 to 5 prime oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3 prime end, moving continuously towards the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5 to 3 prime, called the lagging strand, and is completed in a segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5 prime end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides. The DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, two identical copies of the DNA molecule result.